Hi everyone, in this video we're going to talk about how urine is made in the nephron. So the first step is filtration. This right here is known as the afferent arteriole. The afferent arteriole is what brings the blood supply into the nephron. And as you can see right here, the afferent arteriole turns into this network of capillaries, and I already have the arrow drawn, which is known as the glomerulus. And now, there's rather high pressure in the afferent arteriole and the glomerulus, and that will cause substances to leak from the glomerulus and into this spot right here, which we call the Bowman's capsule, or sometimes the Bowman's space. And what kind of substances will be filtered here? Well, small substances. And the reason for that is the Bowman's capsule is lined by a very tight layer of cells, which are only going to allow very, very small substances to pass through. So what will pass through, for example? Water will pass through. Glucose is small enough to pass through. Amino acids, I'm just going to abbreviate that, AA''S. Ions will pass through, sodium, potassium, chlorine, etc. And there are certain substances that should not be passing through into the Bowman's capsules. And that would be red blood cells. They're too big to land up there. And most proteins will not be able to pass through. Proteins that are smaller than 30,000 Daltons will pass through, but most proteins should not pass through. And the substances that pass from the glomerulus into the Bowman's capsule form what's called the filtrate. And the filtrate is what will eventually become urine, but of course we didn't get to that part yet. So the filtrate, after being in the Bowman's capsule, passes on through into the next part of the nephron, which, if you remember from our last video, is the proximal convoluted tubule. And what's going to happen here is known as reabsorption. A lot of the substances that we just pulled out of the blood, well, they're actually going to be thrown right back into the blood. And how's that? So surrounding this entire tubule is a network of capillaries, which I didn't draw before because I think it just makes things look a little messy. But surrounding the tubule are the peritubular capillaries. And what's going to happen here in the proximal convoluted tubule is that certain substances will be reabsorbed from the filtrate back into the peritubular capillaries, and from the peritubular capillaries they're going to be reabsorbed into the bloodstream. So what will be reabsorbed here? Well, we want to keep all of our glucose. So glucose gets reabsorbed, in fact all of it. Some of the water gets reabsorbed, in fact actually like 60% of the water gets reabsorbed, and some ions will get reabsorbed here. Okay, we move on from the proximal convoluted tubule into the next part of the nephron. So we're passing through, you can follow the arrow, into the loop of Henle. That's this loop dipping into the medulla. You can see right here that the loop of Henle is in the medulla. And the loop of Henle has two parts. There is this part right here. That is the descending loop of Henle. And over here, where the filtrate will go up, is the ascending loop of Henle. And something very interesting happens here in the loop of Henle. So the ascending loop of Henle is not permeable to water. And that's a little bit of a problem for us because we actually want to reabsorb a lot of the water that's in the filtrate. We don't want to lose all of it. So although it's not permeable to water, it is permeable to ions. And what's going to happen is the ascending loop of Henle will actively transport ions into this space right here that we call the medullary space, or sometimes it's referred to as the interstitium of the medulla. So ions will be actively transported from the ascending loop of Henle into that medullary space. How does that help us? Well, The descending loop of Henle is not permeable to ions, but it is permeable to water. So the descending loop of Henle is permeable to water. What's going to happen? As you know from how osmolarity works, wherever you have a place that has a very high concentration of ions, water likes to follow. So water will be pulled out of the descending loop of Henle into the medullary space. From the medullary space, they will land up either in the peritubular capillaries or in some other capillaries, and eventually they're going to land up back in circulation in our blood, which is exactly what we want. So, by increasing the osmolarity in the medullary space, we pull water out of the filtrate. And what we just described here actually has a name, and that is counter current multiplication. Countercurrent just refers to the fact that the filtrate is going in opposite directions in the loop of Henle, and one side is going down, the other side is going up. And multiplication refers to the fact that we've actually amplified or multiplied the amount of water that we reabsorb, which was our goal. Okay, moving along our nephron. From the loop of Henle, we will go to the next part of the nephron, which is the distal convoluted tubule. Distal convoluted tubule, and we're going to talk about it together with the collecting duct because they kind of act in similar ways. Whatever happens in one, a lot of the same thing happens in the other, so we'll discuss them together. This is the collecting duct. And two things happen here in the distal convoluted tubule and the collecting duct. The first is secretion, which is that substances from the peritubular capillaries, in the blood, will be actively transported from the blood into the filtrate. This is different than filtration. Filtration depends only on the pressure in the afferent arteriole and the glomerulus and on differences in concentration, but secretion requires active transport. We need ATP to transport substances from the blood into the filtrate. And this acts as a backup mechanism to the glomerulus because maybe in the glomerulus we didn't exactly reabsorb everything we were supposed to, so the distal convoluted tubule and the collecting duct act as a backup to that in their ability to pull substances out of the blood so that they go into the filtrate. And then the next thing that happens in the distal convoluted tubule and the collecting duct is concentration or dilution, which is our urine. Either we're going to make it very concentrated or very dilute, and that will of course depend on what we want to do. If you're very dehydrated, you will want to hold on to your water. And in that particular case, a hormone, ADH, or anti-diuretic hormone, will be released. An anti-diuretic hormone does the following. Normally, the distal convoluted tubule is not permeable to water, but in the presence of ADH, it becomes permeable to water. So if you're very dehydrated, your posterior pituitary gland will release ADH, and that will cause the distal convoluted tubule to reabsorb a lot of the water. And in that case, you're going to make very concentrated urine. There's a different hormone that can act on the distal convoluted tubule and the collecting duct, and that hormone is aldosterone. Aldosterone will cause you, that was ADH, aldosterone will cause you to make dilute urine. And how will it do that? Aldosterone will actually cause your distal convoluted tubule and your collecting duct to reabsorb more salt. And this will happen in a time that, for example, you have very low blood pressure. Right? As you know, salt causes, high salt causes higher blood pressure. If you don't have a lot of salt, you'll have low blood pressure. So if your blood pressure is very low, you're going to want to reabsorb a lot more salt. And in that case, you're going to make dilute urine.